Welcome to the second part of using BLAST to identify homologous proteins. If you remember, in my last video, I have gone through the basic information where you could use the BLAST to find, for example, organism name and strain name, chromosome location, the biological function of the protein, the length and molecular weight of the protein, and the conserved region or domains. So I have shown you where you can find this information in BLAST. Now, moving forward to the actual question that we can identify homologous proteins. So, in this part, using the amino acid sequence of HOG1P as a query, try to find homologous proteins in genome of Baker's yeast, which is Saccharomyces cerevisiae. The taxa ID is also given here, this one, and fission yeast, which is whose scientific name is Schizosaccharomyces pombe, and the taxa ID of this fungal species is 4896. This one. So we will be performing species-specific BLAST-P searches against RefSec proteins database, and it's important that we should switch from our NR database to RefSec protein database while we will be using the BLAST. And so, but what we will be achieving, basically, we do some answer to these questions. For example, we will be describing the results of search against Saccharomyces cerevisiae genome, how many hits with a score above 200 we will get. So let's go to the BLAST. If I just take you to the, or if I just start from the previous part. So if you remember, I have shown you in first part of this series or this video, uh, I just said to you that you select protein here, use your accession number, click enter or search. So you will be getting this basic information which I covered in the first video. And if you click here in FASTA, then you will get this sequence of amino acid. It's up to you whether you want to use this sequence or you can use this accession number, this one. Either way, we can reach to the conclusion. So let's start our search on BLAST. So I just click on NIH, and then on the right side you see BLAST. I will click on BLAST, and since it's a protein sequence, we will be clicking on protein BLAST. So here it's up to you. You can either enter the amino acid sequence that I showed you in the FASTA format. You just copy from there and paste here, or you just write the accession number. So I'm just using accession number. The job title automatically pops up, but if you want to change, you can change it. So if I want to change, I can change it like question 1. That is okay. Here, since we are required to find for the Saccharomyces cerevisiae and Schizosaccharomyces pombe, so let's just copy this scientific name and put it in the organism field. It automatically populates with different organism lists name, but we will be using this with this specific taxa ID. And then we will add another organism. I will just click here on add organism. Now I will copy this second organism of my interest. I will just enter here. Okay, and now I click here with the specific taxa ID. Now that we have all our organisms here that are required from our assignment or our question or from our practice, let's have a look on this part. We have already selected standard database. That is fine. This is also fine, BLAST-P protein. And I will select this. So we will be getting the result in the separate window. But before moving further, first we will, I have just pasted these two organisms, so it will save our time. So instead of going for the first organism and then for the second, what I will do is just I check this exclude, So it will only now compare Saccharomyces cerevisiae with this taxa ID to this accession number. And then I will just click blast. It will open a new window. Meanwhile, it is doing its job. As we can see, the job has started. I will go back and now I uncheck and check this one. So what will happen that it will compare Schizosaccharomyces pombe against this accession number result. So, and remember that we need to change from non-redundant protein sequence to reference protein, which we didn't before, by the way. So what you have to do, you will again paste the organism and you will hit blast. And now just we will wait. Again, we repeat this for the Saccharomyces cerevisiae because we have changed our database from non-redundant to reference protein. So I just copy the name of my organism of interest. I will just paste here and click here. I will exclude because I'm running the job for this, so I don't need this time. I just need this time Saccharomyces cerevisiae and just I click blast. Actually, I close this window because I didn't choose the proper database because I was choosing this non-redundant, but we need actually reference protein database. So you can see that the job title is now question 1. Meanwhile, we will be waiting for the results. Oh, okay, now there is one result that has been popped up for Schizosaccharomyces pombe, as you can see here. It will take some time to appear my result. Okay, so now if we check for Schizosaccharomyces pombe, we have got the result. And also if we check for Saccharomyces cerevisiae, we also got the result. Now what? Now what we have to do, we have to describe the result of search against Saccharomyces cerevisiae genome. How many hits with a score above 200 colored in red were found? All right, so if we click on graphic summary, and here you can see different lines in different colors. What they mean is that its alignment score, here you can see that it is orange reddish color which shows more than 200. So the alignment scores with more than 200 are shown in this orange or reddish orange lines. So how many lines are here? 1, 2, 3, 4, 5, and 6. So there are 6 hits which have score more than 200 alignment scores. So we can just write here that we got 6 hits whose score was more than 200. And how many in case of Schizosaccharomyces cerevisiae? So in this case, there are only three hits for Schizosaccharomyces. We only got 3 hits more than 200 alignment score. Okay, the next part is provide names of the high scoring hits. Short names like three-letter abbreviations with a numerical index are sufficient. For this part, first we will solve for the Saccharomyces cerevisiae. So if you just hover over the first line, you can see that there is a three letter in the first line, like mitogen activated protein kinase HOG1. So HOG1 actually is required here, that we will write because it is abbreviated HOG1. What is the next one? So the next one is KSS1. KSS1, the third line. SLT2, the fourth line. FUS3, and so on. So you will write remaining two names. And for Schizosaccharomyces pombe, so the first line STY1, because we are solving for both organism. So I will just STY1, sorry, STY1. The second one SPK1, SPK1, and the third one PMK1, PMK1. All right, so and you can finish on your computers the next two names for Saccharomyces cerevisiae. Then the last part is, what do they have in common? What are their biological functions? So if I just go back here to the window of Saccharomyces cerevisiae, so what are their functions? So functions you can find here in description. Mitogen activated protein kinase. This is actually the function of this protein, and you can just copy this and paste it here. This is the function, and the commonality is that they are MAP kinase proteins, or their function is this one. And what about Schizosaccharomyces pombe? There also description is this one, since we are comparing it against because we have provided the accession number of Saccharomyces cerevisiae. So actually all the proteins which are similar or aligned similar to Saccharomyces cerevisiae and Schizosaccharomyces pombe, of course their function will be similar. So this is how you find the information and how you use the BLAST to identify homologous proteins. They are homologous because they share the same function. You can also write the same answer for this Schizosaccharomyces pombe section. Do the best hits identified in Saccharomyces cerevisiae and Saccharomyces pombe have the same biological function? Yes, they have the same biological function. Yes, they have demonstrated, they have shown similar functions. That is MAP kinase, mitogen activated. It was here, protein kinase. So that's it. So thank you very much for watching this, and I hope that you will keep liking and subscribing the channel for getting more knowledge. Bye bye.